So good afternoon. Welcome to this second meeting of the International Postgraduate Course on Mechanisms of Metabolic Disorders. This is actually the first time that we have the scientific sessions. We have three speakers today: Mark Henson from Southampton, Paola Casunello from Chile, and Amanda Sferuzzi-Perry from Cambridge. And since our time is limited, I would immediately start with the first speaker, Mark Henson from Sutton. He will give an introduction to the developmental origin of health and disease. He's the founder and past president of DOHaD, and his research concerns how nutrition and the environment affects health across the lifespan, starting from conception. Basically, his work crosses between the development and the evolutionary biology and also social sciences. So Mark, go ahead. Please all mute your microphones in order to avoid echoes. Go ahead, Mark.
Great, thank you very much indeed, Harry. And good afternoon, good morning, good evening, everybody. So it's a pleasure to start by introducing DOHaD, the Developmental Origins of Health and Disease, to you. I'll try to be as brief as I can. I do have some slides that are quite dense with information, so I'm not going to spend a lot of time going through them, and I'm hoping that you'll have the slides available afterwards. Why won't this advance? Some reason, there we go. So when you ask your colleagues and friends what the biggest health challenges we face in the world today, many people will say, well, probably COVID, isn't it? But the truth is that actually COVID, along with other infectious diseases, account for far less in terms of global mortality than the non-communicable diseases, or NCDs, cardiovascular disease, diabetes, some forms of cancer, chronic lung disease, the list goes on. They account for more than 70% of deaths every year around the world. And actually, the 80% of those deaths sadly occur in low and middle income countries, which can't afford the preventative measures. So we're going to be talking about the NCDs and where they come from. And when you ask, again, your colleagues and friends that, once you've explained what they are, they often say, well, isn't it a combination of bad genes you've inherited from your mother and father, plus lousy, unhealthy adult lifestyle? There's some truth in those things, but it is not the whole story by a very long way. And we need to spend some time in thinking about DOHaD on why that's wrong and what the real answers are. Let's start with this question of the genetic predisposition. Of course, when the human genome was published around the turn of the millennium, we thought that we would find the genes that confer risk of these common diseases. But it wasn't long into the millennium when it began to be clear that that wasn't the case. And so here's a review from Nature in 2008, where the author Mayer says, the case of the missing heritability. When scientists opened up the human genome, they expected to find the genetic components of common traits and diseases, but they were nowhere to be seen. Where'd they gone? They're not in the safe. Perhaps it's the burglar leaving here. So quite clearly, there was a problem that the single gene inherited causes of these diseases were not to be found. Following that, of course, we've all thought, oh, well, these are polygenic diseases. They involve multiple numbers of genes. But unfortunately, even that, when the large numbers of risky genes have been put together, the story still doesn't stack up to a very large extent. People have tried even up to 4,000 potential genes, single nucleotide polymorphisms usually, and linking them to risk and disease. And here's one paper, which actually is now rather old, it's from 2018, but it just shows just part of the story, where they looked at a polygenic score for common diseases, and I underlined the conclusions which related to a threefold greater risk, so a very substantially greater risk for coronary heart disease, for atrial fibrillation, for type 2 diabetes, and so on. And you can see that the proportion at the population level accounted for by these fixed genetic effects is very small indeed, 8% at the maximum. So what's missing? What's missing is our early life in the story and the early life environment. And most of this we owe to Professor David Barker, who worked actually in Southampton, where I work now, and he sadly died some years ago. Barker didn't invent DOHaD, the Developmental Origins of Health and Disease. The idea actually goes back at least to Sigmund Freud at the end of the 19th century, even earlier than that, really. But it was Barker who made the world realize how important it was because of some dramatic data that he had from men, in this case, born in Hertfordshire, a county north of London in England, where he had data on what they died of as middle-aged and elderly men, 61 to 70 years, in relation to their birth weight all those years before. And he showed the dramatic inverse correlation between birth weight and hypertension, type 2 diabetes or impaired glucose tolerance, and the metabolic syndrome. It was a graded effect across the entire normal range of birth weight. So not just pathologically small babies having the risk, all babies had a risk, but it was graded according to their birth weights. There's something interesting going on here at the end, as if there's a high birth weight story too, and I'm sure we'll come back to that in this afternoon's talks. Well, of course, anything found in Britain at that time, this was in the 1980s, 1990s, was not really given a great deal of credence until it had been repeated in a very large study in the US of A. And here, nearly 23,000 men from the US's health study were examined. There were a much wider range of ages, but the same story, birth weight inversely related to hypertension and diabetes. So something is going on in early life, which affects this surrogate birth weight, and then affects all these years later the risk of these diseases. This became what we now think of as DOHaD. We can put the picture together in terms of two critical concepts, really. One is that this plays out across the life course, with a particular emphasis on environmental factors in the first 1,000 days. And the second is the idea of mismatch. So to start with the life course, it's clear that the risk of these diseases increases in a non-linear way throughout life. It's non-linear because plasticity gets less as we get older, and the exposure to poor diet and possibly smoking and excessive alcohol and lack of exercise and so on builds up in its effects on our bodies. We start at different places. People who start with a good start to life in terms of their early development and a higher birth weight in Barker's data will follow a slower trajectory of risk throughout life. Those who have a poor start to life will have a steeper trajectory throughout life. But it's not just this trajectory that matters, because coupled with it is the programming effect on physiology that you'll be hearing about later, which affects our responses to the challenges of life. So if you start and have a bad start to life, you're more likely to be mismatched to meet the challenges of life. And so the impact of that in increasing risk of NCDs will be greater for the red curve than for the blue curve. How does it work? Well, I'm sure you'll be hearing throughout the course and through this afternoon about epigenetics, the processes by which environmental factors affect the inherited genes, not changing the structure of these genes, but changing how they work and therefore how the phenotype of the developing baby actually develops. And I wanted just to show you a couple of bits of data from our recent studies in Southampton, the first link with Lucilla Poston's group in London, and the second here entirely in Southampton, which just show, I think, some of the potential for epigenetics across the life course trajectory of risk. In this paper from Eliane Thun et al., we showed that DNA methylation signatures, and most of epigenetics is involved DNA methylation, not because it's the most important thing, but it's the most easily studied. DNA methylation signatures in cord blood, so at birth, are associated with birth weight. Well, that's following Barker. And we found that they are enriched for differentially methylated CpGs, which had been previously shown to be linked to maternal high blood pressure or preeclampsia, smoking, or low folic acid intake. So you can begin to see a story emerging here that aspects of mother's lifestyle and behavior and diet are potentially affecting the development of the baby. We can measure it through these CpGs, and they are CPGs that are linked in some way to birth weight. Well, that's all very well, but does it matter? Well, we think it does if we look at the second paper here, which shows the influence of, of maternal lifestyle and diet on the perinatal DNA methylation. That's kind of this story. But then we've shown that we can follow this through and link it significantly to childhood arterial stiffness, actually, and indeed fat mass in these children 8 to 9 years later. Arterial stiffness is a good measure of a structural change in the vasculature set up in early life, which affects later risk of cardiovascular disease. It's an early marker. So we begin to see a story of mum's behavior and environment affecting the child, projected through, at least in the early years of childhood, into some risk factors here for cardiovascular disease. And if we put that onto the story of the mismatch scenario, then what it's saying is that really the risky lifestyle factors of women, and I'll say something about their partners in a minute, pass the risk of these diseases back to the next generation. So we genuinely have a life cycle story, as well as, of course, the life course for individuals. Well, it's great to have ideas like that and draw little diagrams, but is there any data that actually supports it? There is. This data from Israel, a very large 1.7 million, I think entirely men, shows just that sort of graph, but it's different in two respects. One is that we're not now looking at risk of disease, we're looking at cardiovascular mortality. And the second thing is that it doesn't start at birth. This particular data starts in adolescence. So they're measuring mortality from cardiovascular disease in relation to BMI of these individuals measured when they were adolescents. And as you'd expect, those who have the highest BMI are the ones who follow the highest trajectory of risk, and those who have the lowest BMI follow the lowest trajectory of risk. But clearly it's graded across the entire range of BMI and therefore risk 45 or so years later. So there's no safe place. It gets riskier the higher the adolescent BMI and the steeper the trajectory followed. So we can begin to see how such processes and the mechanisms that I've alluded to really do affect the entire population to greater or lesser degree, and hence why it might be that 70% of the deaths globally are due to NCDs. The critical thing is that it starts early. In fact, there's no difference in risk in these adolescents, but plays out over time. And we can go back from adolescence into childhood and cardiovascular risk. And this paper, which it's worth looking at, it was literally published early this week, looks at cardiovascular risk factors and adult cardiovascular events in a range of cohorts brought together in the States. And I won't go through it in detail, but you can see childhood risk factors here, coupled, on their own or coupled with adult risk factors for fatal events, cardiovascular events, or non-fatal events. And the story is that you have a childhood effect, you have an adult effect, and putting the two together, you get an even greater effect. And so the story of the mismatch and the additional effects of adult risk factors built onto early life risk factors does seem to be playing out in these very large cohorts. Well, what about the right-hand end of Barker's graph? And what about the problems of this kind of story, the potential passage of obesity and the potential pathology with high fat from one generation to the next? We know from now old data from the Aberdeen study from Rebecca Reynolds et al. That this maternal BMI does indeed confer effect across generations. In this study, they used a Kaplan-Meier plot to look at mortality in the population over 60 to 70 years, starting at birth here, in relation to maternal BMI before and then in early pregnancy, allowing for other risk factors such as smoking, educational status, etc. Women who were obese had offspring who follow this orange line, who have mortality earlier in life and a greater mortality than the opposite, which is the red line, who women of normal BMI before pregnancy and in early conception. Not a massive effect, but highly significant. But what really strikes me is how early in the life course it starts, at the age of when the offspring are probably in their early 20s. So the same life course story. What sort of things are actually passed across then from one generation to the next? Do we have a feeling for what some of the factors might be? And this again is very recent data from our group using the Southampton Women's Survey, where we looked at diets in women and their families actually before conception, in early, late pregnancy, and then in their infants, 6 months up to 8 to 9 years of age. And what really struck Katie Dalrymple and our colleagues was that you could grade those diets into a score of a very healthy, we call it a prudent diet, or a very unhealthy, imprudent diet. And of course, what makes a healthy diet here and here is somewhat different. But given the scores, they're remarkably constant. Women and children seem to follow very prudent diets or normal diets or very imprudent diets in a remarkably constant way. So does that matter, that passage of diet, dietary quality, if you like, across generations? Well, yes, it does, because if we look at the body composition of our Southampton Women's Survey children 8 to 9 years later, as the diet quality deteriorates from the highest score to the lowest, so the body fat mass of those children 8 to 9 years later is greater, and their muscle mass is less. So they have less skeletal muscle and more body fat. And so two of the risk factors for later diabetes and cardiovascular disease, low muscle mass, high body fat, are already set on trend very early in life and related to mother's diet. We can really test the mismatch idea in this population too, because if we look at fetal growth from rather faster in late gestation to rather slower, and then look at fat mass measured in the children 9 years later, when we put diet on the story, we see how it plays out. Those children who grew rather faster as fetuses in late gestation, the effect of a worsening, unhealthy or imprudent diet doesn't seem to affect their body fat mass at all. So these are not mismatched. But over here on the left, children who grew rather slower in late gestation might have been the low birth weight babies of Barker's data. They're potentially mismatched if they're faced with an unhealthy diet later, and that's exactly what we see, that they are fatter as a group, the worse the diet that they consume as children. Oh, what's that doing? Well, it's there to remind me, and this poster was from my child, as you can see from the beetle's haircut. It's sort of 1960s or 70s, and it's saying to men, would you be more careful if it was you that got pregnant? In those days, it was around contraception, but we can see that in fact now there's an enormous range of data that shows that paternal effects can, via epigenetic changes, affect the sperm and have long-term consequences on the development of the offspring. I won't go through this in detail, and there are many more papers, but just to say that it relates to father's diet, obesity, alcohol consumption, smoking, drugs, even advanced age, effects on the sperm affecting the next generation. And if you're interested, this is a very important burgeoning area of study, which these papers will just give you some clue to. One thing I want to say is that, of course, the kinds of things that I've been talking about so far in terms of diet and so on are just one aspect of the much bigger picture of the socio-economic and structural drivers underlying DOHaD. And I think in some ways we haven't, as a community, made it clear how important they are. Deprivation, low investment in education and healthcare, especially in adolescence, ethnic factors. We think of refugees at the moment very much, don't we? Mental health issues. And I wanted to show you just one paper that shows just how powerful they can be. This is data from England, published in The Lancet last year, where the authors are looking at stillbirth, preterm birth, and fetal growth restriction. These are associated. Preterm birth is associated, as is FGR, with later risk of disease, and of course, stillbirth is a tragedy. But what the authors showed was that if we look at the risk factors for stillbirth, preterm birth, and and fetal growth restriction, a very substantial percentage of those can be attributed to socio-economic and ethnic factors. So when you factor in ethnic group, smoking, and BMI, they largely go away. The red increase with deprivation in each of these is reduced, and the ethnic effects, if you allow for them, are also reduced. So this is a real indictment of not just what's going on in England, but in many other countries too, that we're not really addressing these early drivers of risk, which are to do with the structural, socioeconomic and social justice aspects of our society. Some years ago, the United Nations produced this report, and they began to think about the cost of and the cost benefit of this time in life. And actually, here's one single quote: if investing in women, children, and adolescent health gives a 10-fold return on the health of nutrition of women, children, and adolescents, better education results, workforce participation, social contributions result. So this, in addition to all the DOHaD story, gives this enormous return. A 10-fold return is bigger than anything else that any government can do in terms of interventions. So I can't stop without saying just a word or two about COVID. Here's the story that I've shown you already in a slightly different format. Health, well-being, resilience associated with social capital. We start in life at a particular place, it increases, reaches a plateau, and then declines. The 1,000 days is very important in setting that. In low resource settings, everything is worse: poorer start to life, slower increase in capacity, whether it's skeletal muscle or adverse deposition of fat or cardiovascular structure, you name it, to a lower plateau, which is shorter, and a faster decline. Add COVID into that, and absolutely everything is shifted down. I don't know how big all these effects are, but we're talking just in the UK, in this effect, if you look on a high income setting, of at least 500,000 life years lost over about 1 to 2 years. So an incredible effect on the life course of a population. And this is just data literally from that we've just been analysing, which shows the increase in childhood obesity in children going into primary school and children leaving primary school in England just during the COVID pandemic, a dramatic increase. It's already higher for those who are in a lower socioeconomic group than those in the best parts of the country. But wow, this shoots off to levels we've never seen before in this country, just in literally a couple of years. And there are a range of reasons why that occurs. Some of them are listed on here. I'll leave you to look at this offline if you're interested. All the effects of COVID on maternal, newborn, child, and adolescent health, and the way that they will play out across the economy and across our lives in the future, and of course the lives of the next generation. And so in this paper in the BMJ last year, we argued that actually addressing this is absolutely critical for recovering from the COVID pandemic. That's the slogan of our institute, developing healthy lives. And I think the slide is worth many thousands of words from me. The passage of health across generations throughout the life course and back. So I'm sure I've spoken a little bit too long, Harry. Thank you all very much for listening, and I'm going to stop there.
Thank you, Mark, for this fascinating overview. We have time for a few questions. I cannot see the hands of the students at the moment, so please, if you have a question, ask it in the chat. There's a question from Paola. Thank you very much for your presentation. You showed data that despite having a genetic predisposition to have diseases in adult life with a healthy lifestyle, the development of pathology can be prevented. I'm curious to know if these epigenetic changes can change in the same subject if he makes a change in lifestyle. In other words, if at birth he has a methylation pattern in different genes that predisposes him to pathologies, if he follows a healthy lifestyle later in life, his methylation pattern changes to have a less predisposition to disease. So can you change your genetic methylation throughout life? That's the question.
Yes, you can. It's definitely not permanent. In fact, it's a dynamic thing all the time. To be honest, what we don't know yet is which are the key epigenetic changes that may be involved. If you do an epigenome scan, of course, you're looking at literally hundreds of thousands of epigenetic marks. Some of them look as if they're biologically plausible. Some of them, we just don't know what they mean. They shift in relation to, for example, those trajectories of childhood diet that I showed you. We're literally doing the analysis on that sort of as we speak. So thank you for that. That's a highly pertinent question because it relates to whether these changes not only are biologically meaningful, but can they be used as markers of risk and then markers to track the efficacy of an intervention.
Marcelo, the father's role in epigenetics is also fascinating. In this context, what is the role of the transcriptional factor? It is possible that the target identifies some as a marker. So the question,
transcription.
Yeah, the role of
they certainly play a role, and of course, epigenetic changes can do anything and everything. They can affect facilitating, enhancing regions of the genome, repressor regions. They can interact between different parts of the genome, feed back onto the DNA via non-coding RNAs and so on. There is just such a huge amount of potential for this to produce effects. And of course, we've known this forever because obviously we develop into a whole range of different cells from a single cell with the same genetic material in all our cells. So epigenetics has to be what makes us all what we are and what makes us all different. What we hadn't perhaps focused on quickly enough was how it was important to DOHaD and what that might tell us about interventions.
So one more question. The other questions will be in the chat, Mark, so you can have a look at it yourself. Valentina compliments you with an amazing presentation, and she's asking if there are specific nutrients or foods which can change methylation for the better.
Very definitely. So folic acid is one, and we've known that for a long time because the so-called congenital defects like spina bifida are affected by the woman's, whether the woman has an adequate folate status at conception and early pregnancy. We think that polyunsaturated fatty acids are particularly important, some other vitamins and minerals such as iron and zinc. So we're beginning to assemble now a pattern of healthy diets, which there are no surprises about. We've known it all along, but we're hoping that we can then use that to influence public health and make these things cheaper and more available and more widely known about.
Thank you, Mark. Your presentation is fabulous and your contribution is well appreciated. There are also a couple of more questions in the chat, so maybe you can have a look at it and answer those in the chat. I would like to move on with Paolo Casonello. She's from the Pontificia Universidad Católica de Chile. Paola is trained in perinatology, and also has a Master of Science in Biological Sciences. Research centers on perinatal nutrition, placental function, neonatal adiposity, and gross neonatal immunity and epigenetic mechanisms. And her presentation today will be about early adiposity and immune programming by maternal obesity. Paola.
Thank you very much, Harry. Can you see my presentation?
Yeah.
Yeah, okay. Thank you very much to the organizers, and I was really fascinated by Mark's talk as well. So we're going to keep on moving with the DOHaD concept, and in my case, I'm going to show you data. I'm interested in maternal obesity, so I'm going to focus on this concept, especially because this is a disease with high prevalence, as Mark already outstanded, but especially in Chile. You know, here is Chile and South America, and compared to other countries in our area, we are population with the highest percentage in women in reproductive age having obesity. So this is very alarming, together with Mexico, another Latin American country with high obesity index. In the last National Health Survey that we had in 2017, 38% of women over 15 years had obesity. And you can see there's a fraction of women also with morbid obesity, which has been increasing since the last 2 national health surveys. So in our country at the moment, 1 in every 3 women begins pregnancy with obesity, and 2 in every 3 begin pregnancy with overweight. So this is a very alarming problem. And as you may consider, maternal obesity has a lot of risk factors, not only for the baby, but during pregnancy. These women have a higher risk of developing gestational diabetes, preeclampsia, and to win more weight than what is physiological or expected during pregnancy. There's a lot of alterations in fetal development, mainly malformations, also the risk of fetal death and overgrowth in the baby. And the neonatal period also diseases related to intensive care unit requirement, cerebral palsy, and other very undesired neonatal outcomes. What we're really interested in is how this maternal obesity is passing on this risk for childhood obesity and metabolic syndrome. This very interesting paper published a few years ago tried to describe what this risk was a long life for the offspring. And he studied different factors: maternal gestational, excessive gestational weight gain, or maternal body mass index at the beginning of pregnancy. And as you can see in this figure, which I took from this paper, you can see maternal BMI at the beginning of pregnancy is very categorically important for the development of obesity or excess weight in the offspring. In early childhood, in mid-childhood, and late childhood, let's say mainly in adolescence, you can see over BMI 30, as in this bars here, you can see are a very higher risk for risk 5 times more risk of being obese in the kids. So he showed in this paper very nicely how almost 40%, up to 40% of childhood obesity or overweight could be attributed to maternal overweight or obesity together, and near half of that could be attributed to excessive gestational weight gain. And these are the ladies, as I showed in the previous slide, who gain more weight. So these are the 2 factors that are probably affecting the next generation with obesity. How maternal obesity is passing this risk to the next generation is, sorry, I'm going to try to, no, I cannot, here. Are passing the risk to the next generation? Well, obesity, the pre-gestational obesity, so being obese before getting pregnant, has the risk of any other adult being obese, you know, low-grade inflammation and a serious cardiovascular risk, etc. But pregnancy itself, physiologically, is an inflammatory state, even in a normal weight person. So this adds risk to the woman, mainly increasing systemic inflammation, infiltration of macrophages and adipose tissue, insulin resistance, and endothelial dysfunction during pregnancy, which is what explains why these ladies have higher risk of gestational diabetes and preeclampsia. The placenta is also playing an important role during pregnancy, and you can see very clearly how placental factors, inflammatory mediators, alterations, or better said, increase in nutrient transport in the placenta, oxidative stress, and different kinds of alterations in the structure of the placenta can also lead to adverse outcomes. And finally, the programming of the adipose tissue of the fetus and a lot of metabolic adaptations in the postnatal life and during fetal life, which project to the postnatal life. The slides are not moving here. So women with pregestational obesity have been shown to have an endotoxemia and inflammation. Here's adipose tissue from women who had pregestational obesity at the moment of delivery by cesarean section. They got adipose tissue, and you can see that there is an increase in the adipocyte size and also the macrophage infiltration in the adipose tissue of the moms. And also when they put these adipose tissue in culture, you can see they secrete more inflammatory mediators to the culture medium. Anditoxemia has been described as LPS, mainly in the bloodstream, that is due to an intestinal higher permeability, so a leaky gut, which leads to LPS, low levels of LPS. This is not a sepsis, it's a low-grade inflammation, which is increased by the LPS, and which has effects, of course, on the liver, on the adipose tissue, and also in the blood vessels. It has been described that this LPS levels, increased LPS levels in maternal plasma. This is from normal weight women, and this is from obese women. This is new data from Macarena Lepes in our group, which have not been published yet. And she's interested in study also what's happening in the fetal side of the placenta, so umbilical cord blood plasma. And she also saw that the offspring of the women with obesity have increased levels of LPS. So a previous group has shown that LPS can go through the placenta in animals at least. So we're trying to understand how this maternal endotoxemia is leading to a fetal very early in pregnancy, probably endotoxemia as well. And she could also see that this is dependent on maternal BMI. So women with higher BMI are the ones that have the higher LPS levels in the umbilical cord plasma. So going to the placenta now, how placentas of women with pre-gestational obesity also have an increased inflammatory markers in the placenta. So for the macrophages, for the monocytes and LPS receptors in the cells are higher in the placentas of women with pre-gestational obesity. And you can see here the RNA expression of these markers of CD68, the macrophage activation markers, the LPS receptor markers, and other inflammatory markers, and also markers of macrophage activation and secretion of cytokines in the placenta of women with obesity. Finally, in the offspring, you can see higher fat mass, and it has been shown several decades ago already, but I'm showing you here one of the first papers I could find that show increased skin fold thickness in the offspring of women with overweight and obesity. It doesn't compromise the fetal birth weight, but it does compromise the adipose tissue. At birth, the baby has most of the adipose tissue under the skin. So doing skin folds has been shown to be a good approach and a very easy approach to get an idea of what's happening with the adipose tissue. Here they measured adipose tissue by plethysmography, so PPOD, and they could see that neonatal fat mass was directly correlated with the maternal BMI when maternal BMI was over 30. So when mothers have obesity, the fetus has a direct correlation with the adipose tissue in the fetus, and this doesn't happen when the woman is in the normal weight range. The offspring of women with obesity, we also measured in a cohort in our country different adipokines, for example, leptin, and you can see we corroborated data from other groups showing high leptin levels in the cord blood. Interestingly, this is more important in the female offspring. We also saw, and this has been very controversial, how adiponectin is produced only by the adipose tissue, contrary to the leptin that is produced by many cell types. Adiponectin is exclusively from the adipose tissue, and we see higher adiponectin in the offspring of the women with obesity. This hasn't been completely understood yet, although we know that the kids at 7 years, when they are obese, they have lower adiponectin, which is related to insulin resistance. We didn't see changes in insulin and the cord blood of the women of the offspring of normal weight compared to normal weight women. The mechanisms by which this could be happening is mainly that glucose is freely being transported across the placenta. So there's a lot of GLUT1 transporters being responsible for transport of glucose from the mother to the fetal side of the placenta. These high glucose levels in the umbilical cord go to a normal insulin sensitive baby, which is stimulating the pancreas to produce a lot of insulin, which is going to signal to the adipose tissue to generate more adipogenesis, as it is also doing in the liver. So probably glucose and insulin are together responsible for the higher adiposity in the fetus, together with some fatty acids, which are also collaborating in this process. Going to the immune system, which is the other topic that I think is very relevant to this phenotype, this group in the States showed many years ago that the monocytes, umbilical cord blood monocytes isolated from the fetuses from women with obesity, normal weight, overweight, or obese, had higher levels of interleukin-6 in the blood. And when they got the monocytes isolated and they stimulated them with LPS to see the production of interleukin-6 and TNF-alpha, as I'm showing here as a cytometric analysis, an example of what they would expect to increase the secretion of these cytokines, they saw that the offspring of women that were lean and overweight, the monocytes secreted IL-6 and TNF-alpha as expected. However, the offspring of women with obesity secreted very low, so they had a blunted capacity to produce interleukin-6 and TNF-alpha when these monocytes were stimulated with LPS. So this is kind of an immune dysfunction in these fetuses. And also when you look at the invasion capacity of the monocytes, so the migration capacity when they stimulate them with LPS, the monocytes of the offspring of women with obesity also had a lower capacity to migrate. So not only were they less responsive to LPS, but they weren't able to migrate as expected. Yeah, here you have a positive control of adult monocytes. So in this, if you're interested in this topic, I would suggest you look at this review from a few years ago, very interesting review showing all the factors from the mothers, all the genetic, epigenetic, and metabolic factors that could be affecting maternal muscle, the adipose tissue, her gut, the microbiota, the liver, and how the different factors, nutrients mainly, are being regulated in the transport through a placenta, which is different when the placenta is from a normal weight or obese moms, and how these nutrients are going to the fetus and regulating different aspects of the newborn physiology, especially the adipose tissue. And as we are interested recently in looking at, we have preliminary data showing that the mesenchymal stem cells derived from the fetus, which are the precursors of the adipose cells, are also programmed to respond differently. Mainly, Sofia Villalta is looking at oxidative stress regulators of this adipogenic potential. And as Mark already showed, there's an intergenerational effect. So factors are passing from one generation to the other, and it has been clearly shown how nutrition, lifestyle, psychosocial stress, toxic environmental contaminants, and climate are affecting the generation through different factors, metabolic and also epigenetic factors, going from one stage to the other. So if we could stop this transgenerational effects, it would be very desirable that we could find what are the factors. And as Mark showed, probably nutrition is one of the factors that we've been interested in, looking at different maternal nutritional factors, metabolic factors, and also anthropometric factors in the moms. And this is the research of our team, looking how this can affect the newborn and the growth patterns to the infants. We're looking at placental factors, lipid metabolism, and how nutrients are being transported through the placenta. We're isolating immune cells and progenitor cells from the umbilical cord and looking at the response of umbilical arteries and veins. And recently, since what can we do to do some interventions, lifestyle interventions, like physical activity and diet are not enough. So we need to find what is happening in our population. In Chile, although we have a huge coast, people eat very little fish. The survey of nutrient consumption or food consumption in our country shows that every Chilean eats a few grams of fish every day. So if you look in a week, less than once a week, people eat fish. So, and especially docosahexaenoic, which is DHA for short, is a very important nutrient for all these inflammatory factors in adults. The Mediterranean diet are very useful because of these antioxidants and also fish oil. So what we know today about DHA is that in pregnancy is that it lowers the incidence of preterm and very preterm birth and low birth weight and the requirement of intensive care unit. It increases slightly birth length because it regulates eicosanoids, which are important for endometrial activity and for the contractions in birth. And they also help to have a healthier growth and development of the fetus. And since humans are not able to do very efficiently to elongate the chains of these fatty acids, the N3 series or the N6, we did a collaboration with an epidemiologist, Dr. Maria Luisa Garmendia at the INTA in the University of Chile, where she supplemented with DHA women with excess weight, overweight and obesity with DHA. And DHA can go through the placenta. Here's a schematic of the placenta, the syncytial trophoblast. There's a lot of transporters transporting these fatty acids to the placenta. Placenta is using these fatty acids, but it's also transporting them to the fetal compartment. In this paper a few years ago, they showed how PUFAs, mainly the long chain, over 22 carbons in the fatty acid chains, are being promotively transported or positively transported to the fetus. So they concentrate on the fetal side of the placenta. So in this intervention with a DHA, and we call the EpiFat study, where we're interested in epigenetic factors and adipose tissue in the kids. We had this study with the intervention where they had she intervened 1,000 women, where 657 of them were obese. And a group of them received, in the randomized controlled trial, received 200, which is the normative amount of DHA that every person should eat. So twice a week, a portion of fish, or 800 milligrams in a population with a low DHA in the diet. This is going to be a very interesting effect. And we also had a normal weight control group from our hospital. And there was an evaluation of different analysis during gestation. At delivery, we evaluated the fetus, the newborns, the adiposity, some markers in the cord blood and placentas. And we followed the kids until 4 months, where we invited them again for anthropometry, and we did PPOD on them at this age. What I can show you shortly is that the BMI was, of course, different in the two maternal obesity groups with different doses of DHA. They had higher BMI compared to the normal weight. There was two factors: age in the mothers, which was slightly different, and gestational age, just a few days different in gestational age at birth of the kids. You can see here the glycemia of the mothers was all normal glycemia, but the women with pre-gestational obesity, independent of the DHA supplementation, they had higher levels of glycemia, which is one of the factors that also Mark mentioned and we're interested in looking deeper into. And the second trimester, we did the high glucose, the tolerance test, and you can see here also the glycemia after the tolerance test was slightly higher in the groups with obesity, independent of the DHA. So DHA didn't do any effects on these markers. And in the newborn characteristics, we saw that the offspring of the women with obesity was slightly shorter and slightly heavier than the offspring of the women with normal weight. And also, probably DHA is doing an effect there. The triglycerides in the cord were slightly lower than the ones in the normal weight women, and LDL was higher, and insulin was slightly higher in the offspring of the women with obesity. When we looked at the effect on neonatal adiposity by the skin fold, when you look at the effectivity to see compared to 100 with 800, there was no effect on the adiposity. But when we corrected the efficacy, we corrected by the DHA in the umbilical cord. So how effectively DHA is reaching the fetus, we could see a clear effect on the fat mass in the volume and the percentage of fat mass in the body of the offspring of the women who with the higher DHA levels. And when we followed them up at the 4 months, you can see these children have lower adiposity measurements like subscapular versus triceps skin folds, and also the trunk versus total skin folds was decreased in the group of the offspring of women who received the higher dose of DHA. And we could also see one marker of adiponectin, which was higher in these children at 4 months, which we believe has to do with insulin sensitivity. When we looked at the monocytes in the umbilical cord, you could see that the monocytes of the offspring of women with obesity had higher messenger levels for IL-6, MCP1, TNF-alpha, and also IL-8. And in those of the women with the higher dose of the DHA, these levels were reduced in the basal expression. Macarena wanted to know, this is part of Macarena's work, she wanted to know what happens when you stimulate with LPS. As the previous paper in the American group had shown, the LPS is higher, but there's a blunted response to LPS in the monocytes coming from women with obesity. This is not the DHA supplemented group, only control, normal weight, and obesity, maternal obesity groups. And if the cells were pre-incubated with DHA, there would be a lower response of these pro-inflammatory factors, IL-6, MCP1, and IL-10, which we believe we're trying to understand now. What's the mechanism for DHA to regulate this pro-inflammatory secretion in these cells? We're looking at the receptor of DHA and some of the transporters.
Paola, can you come to a conclusion?
Okay, I'll come to a conclusion. We also did some general, methylomes in these cells. We can see there's a lot of promoters involved in the response in the methylation in the obese versus normal weight, and also in the supplemented groups. There's also broad changes in methylation. And finally, for a discussion, we believe that this is happening very early in pregnancy, probably progenitor cells, which a group has recently shown that the mitochondrial respiratory response of these progenitor cells is altered when the fetuses come. We cannot forget that the preconceptional period, so at least 10 weeks before in the case of the men, or 14 weeks before gestation begins, the gametes are already being produced, the mature gametes that are going to give the fertilization process. So probably we need to go back and look at preconceptional period. So summarizing, the offspring of women with pre-gestational obesity have higher risk of obesity in childhood. Women with pre-gestational obesity have an efficient decrease in offspring body fat at birth and central subcutaneous adiposity at 4 months of life. We can also see a regulation of this supplementation in the monocytes on the pro-inflammatory and immunomodulatory genes. We also saw regulation of DNA methylation. I didn't have time to go into details of that, but we're hoping to publish this later this year. And also maternal endotoxemia could be one of the triggers we believe is programming, probably very early in pregnancy, the adipose tissue and the immune cells. So we believe that at least in our population with a low dietary intake of DHA could be one element to reduce the obesity risk in the offspring of these ladies. So thank you to our team, and thank you very much for your attention.
Thank you very much, Paola. I will pick out one question, and can you then look at the other questions, because the session will automatically close after 1 hour and 50 minutes. So yeah, Amanda can give her presentation. So thanks for the presentation. Pregnancy is considered low-grade inflammation. So would it be interesting to have EPA supplementation in combination with DHA, or would only DHA be enough?
No, absolutely. I agree. We're starting a new RCT the second semester of this year, where we're going to use a combination of EPA and DHA. I completely agree. And probably antioxidants would also be helpful.
Thank you. I would like to move on now to to Amanda. So sorry about that, Paola. But
no, it's okay. Thank you very much.
In the chat. Amanda Sferrucci Perry got her PhD from Adelaide, and after that she moves as far north as she could to the University of Cambridge. The title of her presentation is The Role of the Placenta and the IGF System in the Developmental Programming of Offspring Metabolic Disease Risk. So Amanda, please go ahead. You always have the longest title.
I'm sorry. Can you see my slides?
Yeah, I see it.
Okay, thanks so much, Harry and Luis, for the invitation to present some of the work that we're doing today. As Mark and Paola elegantly highlighted, development in utero is a really important determinant of lifespan, determining perinatal survival, adaptation to extrauterine life, and is coupled to increased rates of morbidity and mortality, particularly if the baby is not receiving enough nutrients during development to grow properly. And indeed, supported by several studies in human populations as well as in experimental animals, we know that a variety of environmental factors in the mother, including obesity, as Paula mentioned, can all affect the way in which the mother supports fetal growth and nutritional supply. And changes in this nutrient provision can lead to permanent changes in the structure and function of individual organs within the fetus that are very sensitive to these alterations, and furthermore can result in organ system dysfunction and an elevated susceptibility to develop diseases in later life, about 50 to 60 years down the track. And these span many systems, from the cardiovascular, the metabolic, the neurocognitive, even the reproductive function. In my lab, we're particularly interested in studying the involvement of the placenta in mediating pregnancy outcomes and offspring health indicators. And this is because the placenta really is that functional interface that connects the fetus to the mother and is in charge of a fetal nutrient and oxygen supply. It's responsible for transporting all the nutrients and oxygen a fetus needs to grow and develop, but it also secretes an array of hormones that are released into the mother, and they alter the way in which the mother responds to insulin so that nutrients are made available for transfer to the fetus for growth. Now, a lot of my lab's work and that of others has really focused on the importance of placental transport function in regulating fetal growth and later life health outcomes. But comparatively little is known about the identity and role of placental hormones in regulating maternal fetal nutrient allocation, fetal growth, and offspring health. My lab largely uses the mouse as a model because it has a relatively short pregnancy and lifespan compared to other species, including humans. Moreover, we have methods that we can precisely manipulate the environment of the mother. We can control the genetics or manipulate specific genes so we can understand their importance in determining placental support and maternal support of fetal development in pregnancy. Moreover, the mouse placenta is beautifully organized into functionally distinct regions which form under distinct genetic instruction. You can see here this image of a wet mouse placenta where the labyrinthine zone, which is this vascularized area, functions in transport, whereas this region here that cups around the transport zone is the endocrine region of the placenta called the junctional zone. These regions can be physically separated, and as I mentioned, they form under distinct genetic instruction, where the endocrine cells form under the expression of this gene called TBPA, which is not expressed in the transport trophoblast cells. Moreover, in my lab, we have ability to assess placental phenotype, maternal fetal nutrient allocation, and metabolism at multiple levels, from the whole body to the molecular levels. And so part of the work we have been undertaken is to use the mouse as a model to identify what factors the placenta produces that allow the mother to metabolically adapt to support fetal growth. One method in which we're doing this is by using a technique called fluorescence activated cell sorting, and two mouse lines, the MTMG, which expresses TD tomato and converts to green when it is exposed to this enzyme called Cre recombinase. And this Cre recombinase is not normally produced by mammalian cells, but has been introduced specifically in transgenic mice and is expressed under that endocrine specific, placental endocrine cell specific promoter, TBPA. And this allows the endocrine cells from the placenta to be isolated because they're green, And we can create very pure isolates of these cells that we can then perform mass spectrometry on. We've also developed techniques to isolate and culture endocrine cells from the mouse placenta. And then we take the cells and the conditioned media to perform mass spectrometry. And we've recently published the methods and all of our data. So I'll only briefly touch upon some of our findings. But basically what we came up with was around 1,100 to 1,500 proteins in our 3 different preparations. We use quite a strict pipeline to convert our proteins to genes, and then only focused on those that were reported to be expressed by the mouse as well as the human placenta, because we wanted to be able to translate our findings to humans eventually. We then further narrowed down our list to about 100 and 300 proteins that are known to be secreted or have predicted domains that allow them to be secreted, of which some of these potentially secreted proteins overlap between our preparations. But we wanted to capture all of these potentially secreted placental proteins. So we combined our different lists to come up with a secretome map of about 319 proteins. We then did pathway analyses and found that the majority of these proteins are implicated in cellular, biological and metabolic processes, which may have particular relevance in the context of pregnancy. And so to address this, we overlaid our list of our secreted placental protein gene list with the gene lists for the placenta from fetal growth restriction, small for gestational age, and other complications of pregnancy like gestational diabetes, which is associated with poor glucose control in the mother. And we found around 1/3 of our placental proteins have been reported to be dysregulated at the gene level in such complications. And I realize that this figure is quite complex, but what I am hoping to highlight to you is that there seem to be specific classes or specific types of placental hormones that are dysregulated in specific pregnancy complications. But overall, these are largely enriched in the human and mouse placenta compared to other tissues, where it's really localized to the trophoblast that is bathed in the mother's blood. Moreover, we found 4 placental proteins that were dysregulated in all of the pregnancy complications that we studied, including this important gene called insulin-like growth factor 2. So we wondered whether we could manipulate IGF-2 in the mouse placental endocrine cells to study its function, but also more broadly, the function of the endocrine placenta in modulating maternal metabolism to support fetal growth. And so we have been manipulating the expression of IGF-2 just in the endocrine cells of the placenta using that TBPA Cree mouse line that I mentioned. And we were particularly drawn to IGF-2 because it's a special type of gene called an imprinted gene. And an imprinted gene means that it is the gene is only turned on if it's expressed either by the mother or inherited by the mother or the father's inherited chromosome. And in this case, IGF-2 is only turned on if it's inherited from the father. So it's paternally expressed. And that's because the mother's copy is silenced by imprinting. Its gene is very highly expressed within the fetus and the placenta in virtually all mammalian species. And in humans, imprinting defects in the IGF-2 locus lead to major growth syndromes like Becker-Wildermann syndrome, which is overgrowth of the baby, and Silver-Russell syndrome, which is severe growth restriction. And as I showed you previously, it's commonly dysregulated in the placenta in several complications of pregnancy. We know that this growth factor shares structural homology to proinsulin and can have a range of growth promoting metabolic effects. We also know its expression in the placenta is very responsive to conditions like obesity, hypoxia, undernutrition, which affect maternal ability to support fetal growth. And moreover, finally, we and others have shown that IGF-2 is really important in the formation and the function of endocrine cells in the rodent placenta. And so we've been using our TBPA-CRE line, which is beautifully specific for the endocrine region of the mouse placenta, to delete paternally inherited IGF-2 or activate normally silent maternal IGF-2 just in the endocrine zone of the placenta, leaving the transport zone, the mother and the fetus unaltered. And the beauty of the system is that we're able to manipulate half or the entire litter in phenotypically wild type normal mothers. So we can look at dosage effects and purely at the effect of our manipulation on the endocrine functions of the placenta. Due to time constraints, I'll tell you about our loss of function model, but I'm happy to talk about the gain of function model in questions or through email correspondence. And just to say, largely our data are not published. So we just wanted to be sure we could alter expression of IGF-2 in the endocrine region. And by qPCR in litters of mixed genotype, we could show that IGF-2 in the endocrine region was significantly reduced, not affecting expression in the transport zone compared to wild type litter mates. And it was also apparent using in situ hybridization, where you can see it's the loss from the endocrine compartment and not the transport compartment of the placenta. Contrary to what we would have expected, we didn't see any change in the size of the endocrine region, but what we did find is dysregulated expression of several key hormones in the dissected transport zone. So in females and male fetuses, loss of IGF-2 from the endocrine cells resulted in an increased expression of this specific enzyme, which would favor the production of estrogen over progesterone. And this is really important because estrogen increases insulin sensitivity and progesterone reduces insulin sensitivity, which I'll come back to a bit later. We also found that there was reduced expression of select members of the prolactin placental lactogen gene family. And this is also important because this gene family is really important in regulating insulin production in the mother in pregnancy. We also performed placental secretome analysis by preparing cultures of our control and our mutant placentas and just focusing on the 55 unique secreted proteins and doing pathway analyses on these. We found that these were indeed implicated in several cellular, biological, developmental, and metabolic processes. And they comprise proteins like prolactins, as I mentioned earlier, pentraxin, and soluble leptin receptor, which have all been reported to be important in controlling insulin production and insulin action. We didn't see any change in circulating IGF-2 in our mice, suggesting, though, to us, in that case, that loss of IGF-2 from the endocrine cells induces placental endocrine malfunction and could be a very valuable tool to study more broadly the function of the placenta in pregnancy. So to address this, we set up crosses of our mutant mice to generate pregnancies where all the litter was normal or the whole litter lacked IGF-2 in the endocrine region. So all of the litter had placental endocrine malfunction. And we compared these pregnant mice to non-pregnant females. And what we found was that there were alterations in maternal glucose and insulin handling. I'm just showing you here the results of glucose tolerance test and an insulin tolerance test performed on non-pregnant mice. So you inject in glucose and you see glucose is rapidly cleared from the circulation. It's also rapidly cleared from the circulation if you infuse in insulin here. Whereas if we look at controlled pregnant mice with control litters, glucose is very slowly cleared from the circulation. It also, insulin is not able to reduce glucose in the circulation as it should when compared to non-pregnant mice, indicating in pregnant mice with normal litters, they've developed glucose intolerance and insulin resistance, which are really important in freeing up glucose and lipids for transfer to the fetus. But when we delete IGF-2 from the placenta to induce placenta endocrine malfunction, we see that these adaptations fail to occur. These mice have similar glucose tolerance and insulin sensitivity to non-pregnant mice. And in line with this, we find that these mice do not have elevated concentrations of lipids in their circulation. They don't increase triglycerides or cholesterol as they should as part of pregnancy. They also have lower concentrations of fatty acids in their circulation compared to pregnant mice with normal litters. We also see alterations in the provision of glucose to the fetus. In mice, we can look at glucose transfer in real time by infusing in radio-labeled glucose and then looking at uptake and transport across the fetus, across the placenta to the fetus. And what we find is that both male and female fetuses from these pregnancies with placenta endocrine malfunction, that they receive less glucose from the mother. They also have reduced glucose in their circulation. And this has consequences for fetal development. We see that these pregnancies have lower numbers of viable fetuses, and weight of both the male and the female fetuses are reduced by about 10%, even accounting for this smaller litter. And although this doesn't sound like very much, clinically this would be highly relevant. But as I started off, we're really also interested in the programming of offspring growth and metabolic function. And so we let litters deliver, and we followed up the growth and metabolic function of female and male offspring that had been supported by control and mutant placentas. Now, remember, these offspring are normal. They were just supported by these altered placentas. And these offspring are born slightly lighter, but they very rapidly catch up their growth, as shown here as a proportion of control weight, where they're not overtly different in body weight by 12 weeks of age. But what I find is so striking is that the metabolic health of the offspring is programmed. When we do insulin tolerance tests on the female and male offspring, you see that they have a diminished ability to reduce glucose in their circulation upon challenge compared to mice that were exposed, that were supported by control placentas, indicating that there is programming of insulin resistance in the female and male offspring as a result of placental endocrine malfunction. And we've done some analyses of the tissues of the offspring, the pancreas, the liver, skeletal muscle, and adipose. And basically what we have seen is that there are sex-specific programmed alterations in insulin production, insulin sensitivity, and nutrient handling in response to placental endocrine malfunction. We also know that there is greater sensitivity of the female and the male offspring to gain adipose tissue or become obese and to become insulin resistant if they're challenged with a high sugar, high fat diet from weaning, suggesting that they have this propensity for metabolic disease, certainly if they're challenged from weaning with an obesogenic diet. And there could also be other impacts on offspring health. In some preliminary experiments where we looked at the female offspring, we found that they underwent puberty earlier and had altered estrous cyclicity when they reached adulthood. In collaboration with Professor Giussani in Cambridge, we've also got evidence that suggests that at least in male offspring supported by the mutant placentas, that they are programmed for altered cardiovascular function with reduced cardiac systolic function, but a compensatory enhanced vasodilatory response in young adult males, suggesting multiple systems of the offspring could be programmed through placental malfunction. And that just leaves me to summarize. I hope I've convinced you that utilizing tools in the mouse, we've been able to show the important role of the placenta in communicating to the mother through the production of prolactin, sex steroids, but possibly lots of other factors to modulate the metabolism of the mother, to aid in nutrient provision to the fetus for fetal growth. Moreover, if we disrupt this process, this can lead to the increased risk of the offspring developing metabolic and other dysfunctions in later life. We're actively working very hard to identify more so what factors are dysregulated in our models that are mediating these metabolic alterations in the mum, how they are brought about, the epigenetic mechanisms by which the programming of offspring may be occurring in a sex-specific fashion, and moreover, to combine our unique tools in the mouse to understand the role of the endocrine placenta in determining pregnancy outcomes in conditions where the environment of the mother is challenged, for example, in response to obesity or hypoxia. And that just leaves me to acknowledge the great people in my lab, particularly those that I've circled here who have contributed to the work, my collaborators in Cambridge and the funding bodies. And just to say, I'm always happy to host and mentor students and postdocs if they're keen to explore exciting projects on the placenta. And thank you for your attention.
Thank you, Amanda, for such a beautiful presentation. I see you already have 15 or 20 students and you want to have more.
I find that having them come from all different parts of the world. I've had people from Chile, from Brazil, from
I know, it's exciting,
which is very nice. So
the effects of placental malfunction, you see them in the offspring. Did you also look at the next generation, so the F2 and the F3 generation, and does this effect of placental malfunction last longer, because there is human evidence for that?
Yeah, so we have started that work, and our preliminary data has suggested that the fetal growth defects that we see in the first generation are carried on into the second generation. So those experiments are quite complex because of the genetic setup, but we think, and that we've only studied that on the maternal line. So when we've had the females mated to wild type males, their fetuses, regardless of sex, are growth impaired. So it suggests that there is some permanence. However, what we don't know is to what extent that is due to the programmed metabolic alterations in those F1 females, or whether it's some epigenetic memory within the developing gametes of the female that then affect the F2. And we still the F3, unfortunately.
Still, it's scary that if your grandmother didn't behave well in regard to her diet or whatever, that we suffer from that.
Yes, yes.
Grace Chung, thank you very much for a great presentation, Dr. Svaruni Perry. I apologize I missed this, but what gestational day were the GGT and ITT performed in pregnant mice, and was this an IP injection? And how was the litter size controlled for the offspring studies?
Yeah, so those are all very important questions. So the data I showed you for pregnancy was all for day 16 of mouse pregnancy, which is the start of the third trimester of pregnancy. We chose that time point because we know that's when the endocrine region of the placenta is largest and the mother is most glucose intolerant and insulin resistant compared to non-pregnant mice. We fasted the animals for about 4 hours and we did IP glucose or insulin injections. We controlled for litter size in our offspring studies by so we allowed the litter to deliver naturally, and on day 3, we standardized our litters to 3 female and 3 male pups as far as possible. So we were able to control litter size variability through standardizing the litters soon after birth.
Dr. Svaruni, what is known about growth disturbances in offspring of parents with Beckwith-Wiedemann syndrome? And now I hope, Amanda, that you know what the Beckman-Wiedemann syndrome is.
Yes, yes. So Beckwith-Wiedemann syndrome is an overgrowth syndrome caused by overproduction of IGF-2 through loss of imprinting. Right. So in terms of the offspring of the Beckwith-Wiedemann syndrome, I don't know if anyone's really studied that. I mean, those individuals are prone to metabolic tumors, and they can often have shortened lifespan. I don't know what impacts it has on fertility, but I should certainly look at that myself. We do, however, know that in women that are normal but carry a baby that has an imprinting defect, like in IGF-2, like a Beckwith vitamin, that those women tend to have elevated glucose concentration. So suggesting that there is some communication of the fetus with the mother. But yeah, I don't know if I really answered your question about the offspring of the Beckwith vitamin syndrome children, but I don't think that data is out then.
Okay. So, any other questions for now, or maybe for the other panelists? Yeah, Amanda, I have one more question. You researched so many biomarkers in the placenta. So the billion-dollar question: what is your top 3 biomarkers for preeclampsia and fetal growth restriction in women in the early stage?
I mean, others have shown things like SFLT and S-endoglin.
No, yours.
Not in ours, but that could be related to species differences because we started with the mouse rather than the human.
Yeah.
My short answer is I don't know. I think it's not going to be any top 3. It's going to be more than that. And we know that, these factors are interacting together, right? So I think what ideally would be great is to have a panel of markers of which then an algorithm calculates risk with looking at all of those panels markers in light with the conditions of the mother, her BMI, her and her other risk factors to give a more informed risk factor for the diseases.
Yeah, and it might not only be proteins, but maybe also methylation products in serum-free DNA
and shed exosomes. Yeah, exactly.
Let's see. I think there's
another question from Augusto Rojas.
I have another question. Were these offspring from first pregnancies in the mother, and do you think there might be differences with increasing number of pregnancies at them experiences?
Yeah, so that's something that so these were all first pregnancy mothers. We're just starting to look at second pregnancy mothers. Interestingly, I didn't go into the details of the gain of function model where we express IGF-2 to increase endocrine output. But interestingly, what we find is that those offspring are also programmed, but also there is memory in the system, and the mums are programmed to have glucose intolerance 8 weeks after a GDM-like pregnancy. So it suggests that in some states, the effect might magnify in subsequent pregnancy, but we don't know because we also know that the first pregnancy is often the greatest challenge for the mother. And so it might be that there is some memory that allows her to better adjust to a subsequent pregnancy, even if there was genetically induced placental endocrine dysfunction.
Thank you, Amanda.
Harry?
Yes?
There's another question from Augusto Rojas Martinez. I don't know if you took it.
I don't see it. Okay, go ahead.
Isn't here? I said, Dr. Esperlucci Perry, what is known about growth disturbances in offspring of parents with...
Yeah, we had that question.
Wait a minute, you had already, okay.
Yeah, we covered that already.
Right, I think it's time to finish now.
Yeah, I think it's time to finish. Thank you very much for the speakers. I learned a lot. These were fascinating talks, and also thank you for the audience for so many questions. So this was really a good start off of the MMD course. Luis, do you have any announcements to make for the next session?
Yeah, this next session is on Thursday, not on Friday. Please make note of that. And you have to register again through the same link, because every session is kind of different, so to avoid other people coming into the same link. And thank you again to Mark Henson, Amanda, and of course Paola for the nice talks. And remember that we just sent an email to students regarding the possibility of writing a manuscript as a product of this course, also with some of the lectures. So some of you have already replied, and please check your email because it's a very good opportunity for all of us. Thank you very much.
Thank you very much and enjoy the weekend. Bye-bye.
Thank you.
Thank you. Bye.